So TB is quite a problem. 10 million people die each year, of which 1 million are children. 2/3 of all cases occur in only 8 countries, of which we are one. So countries like Britain and Germany have about 10 cases per 100,000 people each year. We have more than 600. In fact, in the Western Cape, we probably have close to 1% of our population being diseased with TB. Of the 1.3 million deaths, about 300,000 are also HIV infected. And if you look at the context, 3,650 patients per day die of this preventable, treatable disease. That's the equivalent to 18 Boeing 737 Maxes going down each and every day. The really frightening thing is also the increasing emergence of drug resistance. We are now dealing with untreatable forms of tuberculosis. And for the healthcare professionals, that is quite a risk. Nurses and doctors who get infected with such a strain and become diseased are virtually not treatable. So there are many famous TB sufferers, including Nelson Mandela, and I would like to add 500,000 South Africans each year. Now, TB is a disease related to poverty. Although you can all get it, even if you are very rich, you can get TB. But the TB problem persists because of poverty. And this scheme here shows what the issues are. If we have just under 600,000 people with TB each year, 14% of those never get tested because of our healthcare services and because of the available tests that are often done in a centralized manner. Of those that get tested, about 14% are never diagnosed as TB. So those who actually have TB, they get tested. 17% of those never get diagnosed because they have to come back for the result, or the result is negative the first time around. They have to come back for a second test. Even where I live in Durbanville, when I drive past Durbanville Clinic at 6:30 in the morning, there's a queue from the entrance to the clinic right out to the road. And that's in a privileged area. Now, if you don't work, you don't have income, and you don't have transport, going to the clinic and standing there all day and walking away without a diagnosis and being asked to come back in a week or two is a serious disincentive to making sure that you actually get a diagnosis and go on treatment. Our treatment takes 6 months, tablets every day. So the initial loss to follow up, even after the diagnosis is told to the patient, is another 13%. And a lot of people stop treatment prematurely. So we end up with about half of people who have tuberculosis being cured of this disease. So it's an interplay between social factors, inadequate diagnostic tests, and a failing healthcare system that really leads to this huge problem. Now also, TB is a complicated disease. Here I'm trying to show that this person here transmits the disease. Now, the bacteria are transmitted via droplets. So if I have TB and I speak now, the front row here stands a chance of getting infected. Luckily, infection does not always mean disease. Only about 10% of people who inhale the bacterium actually eventually become diseased, and most do so within the first year of exposure. Then the bacterium can be cleared by your immune system, or it can persist, and we know it can persist for a lifetime. We don't know in what percentage of people who are infected the bacterium persists for a lifetime, but there are case reports of a father who got TB, his son got TB, 20 years later, same organism. People like Professor Gerhard were involved in studies, molecular epidemiology, where we can strain type the actual bug that causes the disease. And the same strain in that father was found than in his, in the son was found than in his father from 20 years earlier. So some people have this bug in them for a very long time, and then if they develop HIV infection or diabetes mellitus, their chances for reactivation of that initial infection is increased. Now, we use immune tests to show that there is possibly an infection. We call it subclinical TB. You can't culture the bacterium, but you use immune tests to show that this immune system has seen this organism before. The only part of this where we can actually culture the bacterium is active disease. When people become sick, they have symptoms, they have signs, they develop fever, night sweats, weight loss, chest pain, cough up blood, active disease. Now we can culture the organism. As soon as we start treatment, the bacteria go away again. And now again, at the end of treatment, we don't know what have we achieved. Have we just suppressed the bacterium back to one of these early stages, or is it actually gone? We don't know. And the only way to find out really at the moment is to wait to see what happens. And in our country, the relapse rate, relapse means reactivation of the original bacterium, happens in about 5 to 7% of people. And in people with HIV infection or with diabetes, it's higher, it's about 10%. But if you wait long enough, about 15 to 20% of people with previous TB get it again. So that means that quite a few people do not undergo sterilizing cure, but only go back to what we call a quiescent phase, where your immune system is needed to suppress the infection. Now, even people with sterilizing cure can get infection again if they get exposed again. So, complicated. Now, there seems to be at last some political move and some political will across the world to deal with this situation. And we probably have the HIV activists to thank for this, because the HIV activists have realized that TB is one of the main issues, and they are much more outgoing and proactive than the TB researchers. You know, the TB researchers are all like me, you know, that's under the clip. So, at the first UN General Assembly high-level meeting on TB a year ago, they decided the following: find the missing cases. Now, we're talking lots of cases. We're talking 40 million cases in the next 3 years that we know are out there that are not being diagnosed. We need to increase the funding by about $13 billion. HIV gets 20 times as much money for TB for research than TB does. So we need a lot more money. So TB is responsible for 2% of the so-called disability-adjusted life years, but only, and 2% of deaths, of all deaths, but only receives 0.25% of the global medical research funding. Okay, now let's address some of the problems. So detecting tuberculosis, those with active disease, is problematic. The oldest test, one of the oldest tests that we still use, and in Africa, it is still the most commonly used test, is microscopy of sputum. And what you need is a microscope. I'm showing an old microscope there, but it really, the test dates from those days. ZN, Ziel-Nielsen staging, the demonstration of acid-fast bacilli in sputum requires a trained laboratory technician, a lot of patients. They need to go through a slide, field by field, to find a few of these organisms. 60% sensitivity. So only 60% of people with active TB have a positive sputum smear. The best test is culture, shown here, and we have now automated culture, quite a fancy system. The bacteria consume oxygen, and there's a quencher in this tube, and if the oxygen is used up, this thing starts fluorescing. And this instrument has trays of these sputum samples in there, and each day the technologists will go and see this and that sample is lighting up positive, so that could be TB. The problem is now you have to confirm that it is TB and not some other bacterium that just consumes the oxygen in there, because this is not a completely specific culture method. So you have to streak out on a agar and do PCR on those colonies that grow. So it's a long process. and it costs at least 450 Rand to do such a test. And the problem is it takes up to 6 weeks to say this is negative. Do you want to wait 6 weeks for your test result? So although it's the most sensitive assay, it has lots of problems. And as you can imagine, this needs to go into a central lab. This is not something you have in every clinic. A great new intervention was the so-called gene expert. It's a PCR, a polymerase chain reaction. This little cartridge there, so you put your sputum in there, and a very clever mechanism moves the sample from chamber to chamber, and the whole PCR reaction with its different temperature settings happens in this tube, and within 2 hours you get a result. But again, nobody has that in every clinic. And I'll show you how well we are doing in South Africa. We have the best rollout of this gene expert instrument in the world. We have hundreds of them. But you can also see that there are large gaps. Now, even where I live, in Durbanville, Fisantekraal is just around the corner, 10 kilometers away from me. There's no gene expert machine there. So if I go to the Fisantekraal clinic, my sputum gets sent away to Greenpoint, to the NHLS. And maybe the result will come back in a week. Maybe it won't. So even in Cape Town, we have problems. Not to speak of this area here and the Northern Cape, etc., where gene expert is just not available. So these guys send their samples to the nearest central lab, and that leads to this huge loss in follow-up. Okay? So can we find better ways to do this? So that's why I work on biomarkers. Now, I'll tell you now what a biomarker is, but we are looking at different types of biomarkers. We want something that helps us with the diagnosis at the point of care. Prevention, vaccine efficacy, and can we identify early in that subclinical phase of TB that somebody is going to develop active disease? Can we identify those people, and can we prevent the disease from going any further and causing lung destruction and being spread to other people? And then also treatment response. We don't know when treatment is enough. When can we stop treatment? Who has a poor risk for poor outcome? So a biomarker is actually any biological measure. Fever is a biomarker. We use the interaction between a macrophage and a T cell, which then leads to the release of inflammatory mediators, proteins. We call it cytokines. We use levels of those cytokines as biomarkers. Now, a biomarker may be an indicator of normal biological processes, pathogenic processes, or the response to pharmaceutical intervention. So we use biomarkers for all kinds of things. One of the most used biomarkers is CRP. It's a protein that is measured to look at general levels of inflammation. So can we do anything with biomarkers? So I want to take you to two studies that we were involved in. The first one was done here in Worcester by SATV, South African TB Vaccine Initiative from UCT, with whom we work closely. They looked at more than 6,000 adolescents in the community. They took their bloods and made sure they didn't have TB and followed them for 2 years. 46 of those 6,000 people developed TB in the follow-up period. And they had samples of these people from before they developed TB. So I was involved in the Gates Grand Challenge study, where we looked at household contacts across Africa, 4,500 more or less household contacts. So these were people who had a household member with active TB within the last 3 months. And we took those household contacts, we made sure they didn't have TB, we took their bloods, we followed them for 2 years, and 80 of them developed TB. So about 2.2% of those household contacts developed TB. And what we did then is RNA-seq. So that's a technique. So you have chromosomes, and the chromosomes have DNA. The DNA encodes for mRNA. So how this works is here you have an exon of a gene, and that gets put together to form mRNA, and that leads then to protein expression. So we believe that the expression of mRNA helps us to determine what the immune response is trying to do. So for RNA-seq, you fragment this mRNA into thousands and thousands of little pieces, and then bioinformatically, because we know what the genome looks like, we can put it together again. On the computer, we put it together again, and then we can say, oh, okay, this gene has so much mRNA that is being expressed. And then we can compare the gene expression in different individuals, in different groups with each other. So it's a well-established technique, and that's what we use to address this progression to TB. So here we developed a model, well actually not we, the guys in Seattle, our collaborators, they developed an interesting way to look at gene expression. Basically they compared just pairs of genes. So on the one axis, you'll see the expression of this gene, on that axis, expression of a random other gene. A red dot means this is a person who subsequently became diseased with TB, and the black dot means this person did not become diseased with TB. And then you draw with fancy statistical methods, support vector machine analysis, you draw the best discriminator line between these two, and that can then go into a model. So here we have such a model, a couple of genes here. Each dot represents one gene, that's another gene, and the line between the two genes indicates whether the person progressed or not. If it's red, if you mark it red, it votes progression. If it marks green, it votes non-progression. You'll see in this model here, there are green and there are red lines. So some of these votes are for non-progression, some are for progression, and then you can set a cutoff and say, okay, 75% of the genes in this model have to vote this or that way before we say it's a progressor or not a progressor. You understand? So we used that, and we have found that this model actually is very robust and that we can use it across many different gene sets out there. Now, in this booster study, the sensitivity of this assay was 66% and 80% specificity. Now you'll say, well, that's not very good, but it's 66% better than what we have now. We have no clue who's going to develop TB. But this test can show more than 60% of people who are going to develop TB. And in fact, if that test is positive, most of them actually do develop TB. So it's a screening test. If we take that model and apply it to our household contact study, in the year before TB develops, we can pick up just about over half. And between a year and 2 years, a bit less, just under 40% sensitivity. But okay, so for some people, even 2 years before they become diseased, we can say, well, you have a reasonable chance to develop TB. Now, that's the best we have at the moment. Now, this is the type of thing that happens in the world now. You are forced by your funder and by the journals to publish your data sets. Initially, we were all a bit taken aback by this. Why must I publish my data set? Because then Nico is going to do his work on my data set. Well, my data set was generated by public funding. So it's nothing but right that everybody should benefit from my or his data sets. So here we look now at different data sets and different signatures and how they perform on our study. And we see here that there are many, in this case 4 different signatures that people have published that actually work quite well on our data set. What we then found is that when you break up our data set into South African samples, Gambian samples, and Ethiopian samples, those signatures start misbehaving. So here you see one of the 3-marker diagnostic signature that only works well in Ethiopia, not in South Africa and not in the Gambia. Here you see a model that was developed on South African samples that only performs well on South African samples in our study. And here you see the Worcester study signature that only performs well on our South African samples and not on Gambian or Ethiopian samples. So this means you don't want a village-specific test, something that only works in Worcester or in Stellenbosch. You want something that works all over the world, and that is a new challenge that we have. So you have to develop your models by including data from all over the world. And that, of course, is expensive. That takes a long time, takes a lot of logistics. But we know now that's what is going to be required. Just want to tell you about these curves. So that would be a perfect, that red line, that would be a perfect test. So what these ROC curves do is they show sensitivity, and on the top you see 100% sensitivity. And on this graph, it should be 1 minus sensitivity, 1 minus specificity. So a test that performs like this is 100% accurate. A test that performs like this is useless. So our test is somewhere between excellent and useless. But it's the best we have. And multiple people have looked at gene expression and proteins. This is more or less what we're going to get. And even if we combine the gene expression with protein signatures, it's not getting a lot better. Okay, but we have tests that can, between 60 and 70% sensitivity, identify people before they get TB. Now coming to treatment response. This shows bacterial burden. This red line here shows our detection limit. So that culture method I showed you can detect above this. So for that culture method is actually 5 bacteria per milliliter of sputum. That's what we can detect. If it's less than that, we can't detect it. That means per milliliter of sputum. So actually, there are a lot of organisms in your lung before you have 5 bacteria per milliliter. So what we think we are seeing with our gene expression is we are detecting these guys here. The immune system tells us, we see this bacterium. We are launching an early response against this bacterium, but you can't measure it yet. Then when they become actively diseased, it's easier because we can culture it, but after 2 months of treatment here, again, we dip under the detection limit and we're flying blind. We know we have to treat for 6 months. If we shorten that period, people relapse, the disease comes back. So we are using biomarkers also to try and tell us more about this period. And it would be great to find a biomarker that tells us, listen, this guy's had enough treatment, we can stop now. And this guy has not had enough treatment, must prolong beyond 6 months, maybe to 8 months. So we used a new technology. It was not that new, but for TB it was new, an imaging technique called PET-CT scanning. PET stands for positron emission tomography. Basically, you inject a dye, radioactive glucose in this case, and you combine that with CT, which is computerized tomography. CT is actually very simple. Each dot here, each voxel, which is a cubic, is a 0.3 cubic millimeter, just gets allocated a density. It can be like air, then it's black. It can be like bone, then it's white. So this is really what a CT is. Now, if we combine this rather vague image of the radioactive glucose with the CT, you get this type of thing. And now we can say, well, if there's glucose there, if it's yellow or red, it means there's metabolic activity. Somebody's eating that glucose. It's usually the white blood cells. And this is another view. So we looked at 100 TB cases and on treatment. All these people were cured at the end of treatment, and you see here at baseline, at month 1, at end of treatment, how the progression looks. And all this red stuff is active disease. That's the heart. Sometimes we manage to cut out the heart computationally, and sometimes we don't. This is not as if these are women and these are men. They all have hearts, but we can't always cut them out. Now, the interesting thing here is we expected at the end of curative treatment, so bacteria gone, everybody would look like this. Cured, some scarring, but no increased uptake. We were wrong, like so often. 62% of people had ongoing uptake in their lungs. They still have inflammation in their lungs. And almost a quarter had new lesions. How do you explain that? That is not just scarring. That could be that the bacteria are actually surviving and are starting new lesions, and the immune system is keeping it at check but cannot get rid of it. So we decided to use that to do RNA-seq experiments, and we came up with our own signature again, with the so-called disease signature, by comparing baseline and end of treatment gene expression, and we use this gene, this disease signature, to check how well that predicts progression in the Worcester study. So now we're looking at people after treatment and comparing that to people before treatment. Now this is for the ACS, the adolescent cohort study, Worcester study. So that's their own data on our treatment response data. You see a symmetry. You see this risk going up, and on treatment, it goes down. The same happens when we look at our disease signature. In the pre-treatment study, it goes up as disease approaches, and it goes away as treatment progresses. So it seems that these signatures can be used at different phases of the infection. That same disease signature was able to show failed treatment at the end of treatment with a very high sensitivity. But even at baseline is again somewhere between rubbish and excellent, that the signature before treatment even is started tells you that this person has a high chance to fail treatment. So one of these signatures that were partly developed on our cohort is called a 3-gene signature, and it was evaluated in in the booster, in our treatment response, and in the Brazilian study, and this performs quite well, 90% sensitivity and 70% specificity. Now, that's pretty good. Now, the guys who made Gene Expert actually now managed to convert one of their cartridges into a point-of-care mRNA detection device. So the whole RNA extraction from whole blood and the PCR can now be done within 2 hours in a little cartridge, which is fantastic. And we are now going to be one of the first to evaluate this. So what about these predictive signatures? So we have done large studies, but now we need prospective studies. Okay, so we had these sample banks, and we went and we looked at our developed signatures. Now we need to take those signatures and do prospective studies. And a very large one, the Cortes study was just completed by UCT, and we were part of that. 20,000 people were enrolled and followed for 2 years. And we'll know the result soon. So if these signatures were positive, people were randomized into either treatment or no treatment and followed to see if the signature actually predicts prospectively if somebody is going to develop TB. The point-of-care devices are on its way. There is one already available. And we need to look at these signatures where they have diagnostic and treatment response utility. Just briefly about another technology to measure proteins in the blood. So it's called Luminix technology. So it's a fancy way of doing this. There are 2 dyes with which beads are coated. And the combination of those 2 dyes can lead to 100 different bead sets. Each color is then associated with antibodies against a particular target. Let's say we have interferon gamma as a cytokine, so this particular dye, combination of those 2 dyes, will be associated with interferon gamma, and you have antibodies against interferon gamma on this thing. Now you have a second antibody that comes in also against interferon gamma, and that has a fluorescent tag on it. Now you run these beads past 2 lasers, and the one laser will tell you, that's the bead for interferon gamma, And the other laser will say, okay, there's so much on it. So you can, in a very small volume, you're talking about 25 microliters. In a very small volume, you can look at up to 100 different analytes in one go. So of course not inexpensive. About 100,000 Rand for a kit that can measure 50 cytokines. But nevertheless, you would not be able to do that with any other technology at the moment. So we use this as a screening test. And we looked at one of our studies where we looked at over 260 TB patients, and we followed them after treatment, and we had a whole lot of cured people, and 12 had relapse, which means they got TB again, and 15 failed treatment. They never converted their sputum. And you see here at baseline, week 2, week 4, and an end of treatment for these groups. And the colors indicate the level of expression of individual markers. Yellow is high expression and black is very low expression. And you see these Persian carpets look different for the different outcomes, right? You see the relapse guys have much more yellow than the cured guys. And you see the failed treatment guys don't really have a progression from high expression to low expression as things go on. And if you look at baseline, you can see there's clearly a difference in the expression of cytokines. These guys who relapse seem to have more cytokines at baseline in their blood than the cured guys or even the failed guys. And if you look at the end of treatment, you can see those are currently, these guys, the relapsers are currently at that stage. They are healthy, but they would relapse later. They would get disease again within a year and a half. And they have, at the end of treatment, more elevated levels of those cytokines than the cured people. So that's the type of thing we're hoping to use to guide treatment. And here we have, again, so some of those ROC curves that shows a reasonable ability to predict relapse with the number of bacteria at baseline in your sputum and also your body mass index. If you have a very low body mass index below 18, that's a bad prognostic sign. And then these 5 cytokines together, they can indicate a high risk for relapse. And we're doing a large study now with China and with the NIH in the US, 5 sites in Cape Town, because we are the only high TB incidence country in the world with PET-CT ability as well. China has a lot of TB patients, but the overall incidence is pretty low actually compared to us. So we're doing PET-CT at baseline to look at extent of disease. We look at the follow-up. Are they getting better? And then we randomize them into a standard 6-month arm or a 4-month arm if they don't have extensive disease and if they progressed as we expected. And this will also enable us to do more of our gene expression and protein signature evaluation. Briefly, a diagnostic. So we led 2 previous consortia funded by the European Union, and yesterday we started with a 3rd one, where one of our students, Novel Chegu, found that a 7-marker signature. So here you see individual markers, right? Levels of individual markers. CRP, the one I earlier mentioned. And you see that the median values are very different. But you see the overlap is impossible. So you can't use any one of those alone to diagnose TB. That's true for all of these. The median value is somewhere, I think it's over here, and that one will be up there somewhere. But the overlap is impossible. If you combine those markers, then you stand a chance. So we developed a 6-marker assay that had 94% sensitivity to diagnose TB. And we put those 7 markers on lateral flow strips. Now, this was the primitive version while we were still trying it out. So you collect blood with a finger stick device, you put it in a buffer, and then you have this lateral flow device with these antibodies against these markers in strips. And of course, this was not very kind to the staff. Infectious risk is high, there's blood all over, so of course that's not something you can commercialize, but it was just proof of concept. And now we have moved to this closed system where a drop of blood is put in here, and 30 minutes later you have an answer. And the answer will be high risk or low risk for TB. High risk for TB means I speak to you, I tell you you have a high risk for TB. I take it seriously, you take it seriously, you come back next week for your confirmatory test with the gene expert. The other people who have a negative test, I can reassure them, you don't need a gene expert. Go home. If you are not feeling better in 2 weeks, then we talk again. So it's just a triage test. We identify those who need the expensive test, who need to come back, and hopefully we'll get more people back that way. So that is the triage approach. So in summary, host gene expression in whole blood seems to track this complicated interaction between the pathogen and the host before they get diseased, while they are diseased, and afterwards. The same signatures seem to be working in different parts of the spectrum. There are promising gene expression tests for these different applications, but the most pressing one would be diagnostic to find those missing cases. Also, the protein signatures are promising. Now, to move from one technology to the next is quite difficult. The point of care, POC stands for point of care. To move from this RNA-seq to quantitative real-time PCR to point of care, the lateral flow cell, that is not an insubstantial step. And we've managed so far reasonably well, but I'm not a salesman, I'm a scientist. We have to show that this works in the field. So this is really a summary of our progress. So we have well-characterized participants. We have blood protein signatures. First, we started with antigen-specific test with culture, 7-day crazy, 7-day culture. Then overnight culture. Then we saw, no, you don't need culture. You can use ex vivo serum, but still these high technology tests. Then we converted them to lateral flow tests. And now we are busy with finger stick blood. And now we have a point of care lab-free finger stick blood-based test that needs to be evaluated. And the same with the RNA diagnostics. We started with RNA-seq. We went to qRT-PCR. Now we are finger stick blood and a point of care test. So we'll see which ones of these pan out. Where are we in the biomarker development? We're probably somewhere here. Clinical performance. If we are successful in the next round, we'll probably get a CE mark and we'll go to impact studies. And if you want to develop a test in TB, you need WHO endorsement. So we're somewhere here, and we have funding and we have partners who will make this possible. These are the people who do most of the work. Our lab team, our clinical team, our statisticians, our quality assurance people. And like always, these type of things can only happen with very large consortia. We led several of these consortia, but all these people are important. And we have multiple consortia for multiple different topics. And that's actually some of the funnest stuff of my life, is actually interacting with people from all over the world. And then, of course, the funders and the participants, particularly the Bill and Melinda Gates Foundation and the EDCPP. It's an EU organization, European Developing Countries Trial Partnerships, and, of course, the university, which makes available a lot of infrastructure and opportunities. And I thank you all for listening.